Bismillahirrahmanirrahim. Distinguished guests, ladies and gentlemen, Assalamualaikum and a very good morning. On behalf of the Genetic Society of Malaysia, I would like to welcome all PGM members and participants to this PGM webinar 2020, one of the annual event of Genetic Society of Malaysia, or known as PGM. PGM has been organizing a PGM seminar annually since 2009 with the theme Towards Excellence in Genetics. Before I pass the session to the moderator, I would like to invite the Honorable President of PGM, Professor Dr. Abdul Rahman Milan, to deliver his welcome speech.
Terima kasih Dr. Azzreena sebagai pengurusi majlis pagi ini. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Bagi pihak Persatuan Genetik Malaysia, saya mengucapkan terima kasih kepada semua yang dapat menyertai webinar kali ini. Sepatutnya seminar ini diadakan pada bulan Mac 2020, tetapi disebabkan oleh masalah COVID-19 terpaksa ditunda sehingga ke hari ini. Terlebih dahulu saya ingin mengucapkan ribuan terima kasih kepada Fakulti Perubatan UKM, PP-UKM, yang telah mengizinkan kami menggunakan fasilitis untuk menganjurkan secara webinar ini yang diselaraskan oleh Puan Sarifah Azwin. Terima kasih kepada Encik Mohamad Noor Isa daripada Malaysia Genome Institute yang bersetuju memberikan ceramah kali ini yang dipengerusikan oleh Esko PGM Profesor Dr. Zilfalil. Terima kasih, dan juga saya mengucapkan ribuan terima kasih juga kepada semua esko-esko yang telah bekerjasama menyediakan ataupun menyediakan webinar kali ini. Jadi saya harap dengan adanya webinar ini kita dapatlah suatu ilmu yang berguna, terutama sekali di atas tajuk Unfold the COVID-19 Pandemic. Yang ini kadang-kadang isu yang sedang hangat diperkatakan. Jadi dengan ini saya serahkan kepada pengurusi majlis. Terima kasih.
Thank you, Pak Rahman. Now let me introduce a moderator for this webinar. The moderator for this session is Yang Berbahagia Profesor Dr. Zilfalil Alwi. Professor Dr. Zilfalil Alwi is a professor of medical genetics and senior consultant pediatrician and clinical geneticist at Hospital Universiti Sains Malaysia, Kota Baru, Kelantan. He received his specialist training in pediatrics, master in medicine, pediatrics from the same university, and later went to University of Glasgow, where he obtained a Master Science in Medical Genetics, and then to University of Aston, United Kingdom, where he obtained a PhD in pharmacogenetics. His research interests includes population genomics, which specialize in genetic diversity of the Malay ethnic group, and genome-wide studies on diseases common to the local population. Professor Zilfalil is the founder and head of the Malaysian node of the Human Variant Project, or known as MyHPV. He is a member of the Board of Directors of Human Variant Project International, and he's the joint chairman for Global Globin 2020 Challenge. He was also appointed as the director of USM Human Genome Center from 2005 to 2009. Professor Zilfalil is the chief editor of several journals, including the Malaysian Journal of Pediatrics and Child Health and Genetics, which is official bulletin of the Genetic Society of Malaysia. He is also the founding and current chief editor of Malaysian Journal of Human Genetics. He is also a council member of the College of Pediatrics Academy of Medicine of Malaysia and founding president of the Malaysian Society of Human Genetics. Without further ado, I would like to pass the session to Prof. Zilfalil. The session is yours, Prof.
Assalamualaikum and very good morning. Thank you very much, Dr. Azzreena, for the kind introduction. I'm very honored to be the moderator for this webinar and also to introduce to you the speaker for this talk. As mentioned by Dr. Azzreena, our speaker today is Haji Muhammad Noor bin Haji Man Isa. Encik Muhammad Noor is a scientist at Malaysian Genome Institute, or MGI, and also the head of Genome Computing Section with specialization in genome informatics and molecular biology. Encik Muhammad Noor obtained his Bachelor of Medical Sciences from Universiti Kebangsaan Malaysia, UKM, and also an MSc in microbiology also from the same university. At the Malaysian Genome Institute, Encik Mohamad Noor works on various genome projects involved, and he was involved in whole genome sequencing analysis, such as animal genome, plant genome, microbial genome, and metagenomics. Among the projects that he was involved with was the rapid whole genome sequencing of the coronavirus genome during the SARS outbreak in 2003. His team managed to sequence and annotate 2 coronavirus within 6 days. He was also involved in sequencing of 2 kelah fish and 26 Malaysian human genomes from various major ethnic groups in Malaysia. To date, he has sequenced more than 20 bacterial genomes and metagenomes from various populations. Currently, he is working on 5 species of Rafflesia, 3 iguanas, 2 crabs, and 2 stingless bee genomes. His latest work involved the sequencing of the SARS-CoV-2 genome from our local COVID-19 patient samples. And today, he will share with us the work that he has done on the sequencing of this SARS-CoV-2 virus genome. And this is a topic I'm sure that many of us as scientists and geneticists are very interested to hear and to know. And Encik Mohamad Noor's talk will take approximately 45 minutes, and there will be about 15 minutes Q&A after that. So if you have any questions for Encik Mohamad Noor, please type it in the chat box. There's a chat box on your screen, and you can post questions there, and I will read it to Encik Mohamad Noor at the end of his talk. So today, the topic for Encik Mohamad Noor's talk is unfolding the COVID-19 pandemic through whole genome sequencing of SARS-CoV-2 viruses. So Encik Mohamad Noor, the floor is yours.
Okay, thank you, Prof. Zilfalil, on a kind introduction on me. Okay, Assalamualaikum and very good morning, everyone. Before we start, I would like for our Muslim friends to recite Al-Fatiha for my auntie just passed away just now. Al-Fatiha. A'udzubillahi minasyaitonir rajim. Bismillahirrahmanirrahim. Okay, I would like to start sharing our work with everybody here on the unfolding the COVID-19 pandemic through whole genome sequencing of SARS coronavirus 2 viruses. So I will begin with introduction to COVID-19 disease, about the SARS coronavirus per se, and also go deeper on the SARS coronavirus 2 genome, and I will also touch a bit on the whole genome sequencing of SARS coronavirus activities that we did in MGI and all over the world. And at the end of the talk, I would like to share how we utilize the viral genome information to understand the COVID-19 pandemic, and also try to make use all the information that we have to understand and combat the SARS coronavirus 2 pandemic. So as you know, the outbreak started about late 2019, which is an outbreak of a novel coronavirus started in Wuhan, China. Originally infected more than 40 people, and most of them developed pneumonia and had contact with the same seafood market in Wuhan, China. At that time, it's not clear how many people were initially infected because the virus causing the outbreak still not known or novel, and thus difficult to diagnose. So everybody still ponder what kind of viruses infected those patients that came with pneumonia and other influenza types of symptoms. So at that time, the virus has been since identified as 2019 novel coronavirus or 2019-nCoV because we don't have a specific name or yet at that time, and later been named as SARS-CoV-2 with the associated disease called COVID-19. So this SARS-CoV-2 sharing about 80% of genetically similar to SARS-CoV during the outbreak of 2002 or 2003, and that's why we call it SARS-CoV-2 because they are sharing the same severe acute respiratory syndromes as compared to previous SARS. So in terms of symptoms and so on, so a bit same as previously described SARS-CoV, but with a different virus. So because of the SARS-CoV-2 is novel and many of the potential risks associated with the virus are still unclear. The disease started late 2019. 2 months later, about 2 months later, more than 75,000 people have been infected globally with 2,000 deaths, and about 98% of the cases at that time occurred in China, but later they also involve more than 25 countries after that. So nowadays, and researchers still looking on the what kind of reservoir that made the transmission of the coronavirus. And they found that this coronavirus originally from bat and transmit to human through intermediate host that still non-conclusive evidence that they say came from pangolin. So at that time, during the early infections as the Wuhan seafood market, so they don't find the pangolin being sold at that market, but other researchers after that discovered that potential pangolin is one of the potential intermediate host before transmit to humans. And later, this virus transmit human to human that can cause a pandemic. Globally, the coronavirus disease, as WHO situation report yesterday, already more than 20 million cases reported worldwide, and more than 740,000 deaths until yesterday. So in our country, we have about 9,114 cases since yesterday, and with 129 deaths. Globally, those mainly now is ranking #1 for the cases is still US, and we are at the, if I'm not mistaken, it's about ranking 58 in terms of number of cases currently. So it's within 6 months, 6-7 months from now. So you see from just one cases reported, a few cases reported at the end of last year, and within 6 months, we got more than 20 million cases reported, but still growing, and it will cost us a bit overwhelming on the outcome of this disease. So it's fatal and cause a lot of burdens to country, burdens to the everyone's that have those diseases. So again, it's infectious diseases caused by newly discovered coronavirus. Most people who fall sick with COVID-19 will experience mild to moderate symptoms and recover without special treatments. For those who clinically healthy prior the infections, but for those who have comorbidity like hypertension, diabetes, and so on, so the risk is more higher. So this how those SARS-CoV spread. So mainly those viruses are caused, COVID-19 is mainly transmitted through a droplet generated when infected person cough, sneeze, or exhale. So these droplets are too heavy to hang in the air and quickly fall on floor or other surfaces. So mainly it will transmit through droplet that we touch using our hands and rub our face and so on. So that's how the, initially being the idea how those virus transmitted. But lately there is some groups that saying that still airborne and so on, but that one still under the debate because a lot of other factors that need to be considered in order to say that there's this virus is airborne. So someone can be infected by breathing in the virus if they are within close proximity of someone who has COVID-19, or by touching the contaminated surface and their eyes or nose or mouth. So that's how the mode of or the way of entry to our body. So through eye, nose, or mouth. And later I will talk on the SARS-CoV virus itself. So again, this virus is discovered 2019 that caused COVID-19 and with the severe acute respiratory syndrome. So it's a single-stranded positive sense RNA genome classified as beta coronavirus, and the SARS-CoV-2 are among the 7 known human coronavirus that infected can cause disease to human. Besides the SARS-CoV since 2003, MERS-CoV that recently discovered somewhere in the Middle East, and now the SARS-CoV-2. So among others, 4 HCOV229E, OC43, NL63, and HKU1 among the known human coronavirus that in fact then can cause diseases to human. Physically, that's how the SARS-CoV virus virion. So they have the spike proteins in this schematic illustration virion. So The red protrusion is defined as spike protein. The gray coating, the envelope actually, composed of mainly of lipid, which can be destroyed with alcohol or soap because the lipid. You can see the yellow deposits are the envelope proteins, the E proteins, and the orange deposit is also membrane protein. So that's how this virus looks like if you look at the electron microscope. They have spike, they have the envelope, and so on. So we go deeper in this SARS-CoV-2 virus. So again, you can see the how the cross section of this virus. So we have a spike glycoprotein, envelope, small membrane protein somewhere at the surface of the membrane proteins, and the hemagglutinins, esterase, nucleoprotein, and the inside those envelope contain the genomic RNA. So, and the SARS-CoV-2 genomes first reported in the GIS8 database. We made publicly available since January 10, 2020. So it contains 29,903 base pair sequences, which distinct 15 ORF or open reading frames, contain ORF 1A, 1B, spike protein, ORF 3A, 3B, EM6, 7A, 7B, 8, ORF 8, ORF 9A, 9B, nucleoproteins, and ORF 10. So it's contain about 20, on average, about 29 to 30 KB sequence. So the organization of major genes or proteins within the SARS-CoV genomes contain a lot of proteases on ORF 1A, non-structural proteins, and so on within the ORF 1A and 1B. So they have the RNA-dependent RNA polymerase within the ORF 1B, and helicase and endoribonuclease, the spike proteins, and so on. So that's how the genomic organization of SARS-CoV-2 looks like. It's very similar to SARS-CoV that discovered 2003. So with a different in terms of sequence only. So nowadays, it's a global effort to sequence SARS-CoV viral genome. Why? Why is everybody try to sequence those viral genome for their own from their own countries' specimens or patients? So a lot of things that can be discovered through the sequencing of SARS-CoV-2 genome. Sorry. Mainly, all those data from the sequencing effort are being shared in Global Initiative on Sharing All Influenza Data database, GIS8. And currently, main database for SARS-CoV-2 genome that's shared or publicly available for everybody. Excuse me. And currently, already more than 20 more than 80,000 sequence already deposited in GIS8, and then can be shared and to all over researchers in the world. And currently, until yesterday, we Malaysian already deposited 108 sequences or genome sequences in GIS8, that sharing our local genome that been sequenced from our various institutions in our country. So out of 108 sequence deposited in GIS8 by Malaysian institution, so MGI contribute 10 at the moment, and we are in the process of finishing another 24 genome soon, because we already have partial genome, just waiting for finishing to get high quality genome before we submit to GIS8 probably next week. And currently IMR already deposited 34 genomes from their samples, and UM already 62 samples, and New Malaysia Pahang 2 genomes deposited in GIS8 for sharing the information about the genome for SARS-CoV-2 information. So why everybody sequence the genome, and what are the genome sequences telling us? So if you remember early or late 2019, early December or late 2019, so nobody knows what's the kind of virus and how to diagnose and so on, so to identify what kind of virus and so on. So with the information about the genome being shared by a group from China on January 10, 2020, so a lot of people start using it to design a specific primer in order to diagnose those viruses. So those genome enable us to use as a diagnostic tools for us to understand what kind of virus, or as a diagnostic purposes to differentiate between those people that infect with COVID-19 and other influenza illness. So the virus genome also telling us about the virus distribution, the migration of the patients, and also the origins of the virus, how those virus migrate from one country to other country and introduce new clusters or something like that. So at the same time, the viral genome also telling us about the lineage, how the nomenclature and the variants and so on. So and it will later be used for, could be used for antiviral discovery and also for vaccine development and testing. So that's the roughly about what kind of information that we can gain from the whole genome sequencing of various samples of this virus. So let's say I'm just to share our initial finding on our first wave of the infection for the case number 1, 2, and 3 that first reported in our country on I think 24th of February 2020. So those family travel from Wuhan to Singapore and then enter Malaysia through Tambak Johor. So that's the travel history for those first 3 cases reported in our country. So we did the sequencing of this first wave of infection in our country, and then with the help of Mamal Kesihatan Awam Kebangsaan Sungai Buloh that provide us the samples in order to understand what kind of migration is it through this virus came from Wuhan or any transit somewhere. So with the genome sequence for these 3, so we see, okay, this our initial analysis. It shows that let's say for case number 1 and number 3, I just omit case number 2 here in this initial because this one we did the 3 for early samples that we have. So we will see here those case number 1 and number 3 from genome for case number 1 and number 3 very similar to the index case from Singapore that reported on 9th of February. So you see here you see the, I'm not sure whether you can see the accession number for the GIS8 EPI ISL 410537. So it's here. So you see this sample number 1 and number 3 actually link with this index case from Singapore, because number 1 and number 3, case number 3 actually wife to case index case in Singapore ISL 410537, and case number 1 actually their grandchildren. So you see it's related to this case and way back to Wuhan. So it's this genome information can be used to track back the migration of where the patient got the disease, so got the virus and so on. So it shows that the information from the genome is traced back to the Wuhan. So where those family actually contract those virus somewhere in Wuhan and travel to Singapore and show the symptoms in Singapore, and some of their family just cross the border. So based on our preliminary analysis, it can show that this whole genome sequencing can be used to track the migration or the origins of the virus. So with this idea also, we can, let's say for the tablet cluster, so out of the blues, we know it came, we still don't know the original of the origins of the virus. If we can get the samples and sequence those sample from the tablet cluster, so hopefully we can use the same approach to track back actually those samples came from which country or which regions or and so on. So at the moment, MGI doesn't have that specimen, so I cannot say anything on it. But we can use the same approach to track those origins of the virus based on this preliminary data. So this just to show you how the genome sequencing can be used on tracking those migration of the virus from across the border. And okay, the other usage of the whole genome sequencing, so we can define the virus lineage of nomenclature. So originally a lot of proposal been proposed by researchers in order to group or classify the virus diversity. So we have to know the diversity of the virus in order to group or try to cluster those virus within the country. And a lot of other people's propose the nomenclature and so on. So the most well accepted nomenclature proposed by Rambau et al. 2020, they are based on phylogenetic analysis. And this nomenclature will help the epidemiology of the disease or viruses. So, and within the GIS-8, they classify those viruses based on the clade. So, they have S clade, L clade, V clade, G, GR, H, and other clades. So it's grouped based on, also grouped based on phylogenetic tree, and then we can see the migration of those samples from where. And at the same time, the GIS-8 also incorporate the lineage developed or proposed by Rambau et al. Based on the lineage A, B, B1, B2, and so on. So it's equivalent to S clade initially developed by GIS-8 team. So that's how we group those. And okay, based on those nomenclature, so distribution of the virus lineage within the Malaysia samples or Malaysian virus genomes. So we have out of 108 genomes that we have, so mainly the lineage B.6 distributed within our country. So these most common countries have the same B.6 lineage from India, Singapore, and Australia. So could be our original samples came through India and Singapore. I think that's initially we got our first 3 cases also came through Singapore. So that's the distribution of virus that within our population in country. So I think a lot more sequence being in the process to be sequenced. So we will see what kind of other lineage that distributed within our country. So, and in okay, in terms of mutations, so throughout the world, so a lot of or majority of the mutation happen at the spike protein with the D614G mutation. So for the spike at the position or amino acid 614, originally from the reference genomes on aspartic acid and suddenly changed to glycine. So out of 80,589 genomes that deposited in GIS-8, about 76% of the genome carry those mutations. and the second highest mutation within the global samples from non-structural protein 12 at the position 323 on the amino acid position 323 that changed to proline to lysine. So that's a major mutation that happened within the samples worldwide. We need to understand those mutations in order to study the effectiveness of the could be antivirals that we can discover later, or could be a vaccines and so on. So the importance of know the distribution of mutations is some kind of at that area. So for antivirals or vaccine. So this among the top mutation happen in the world. And we go back to our country, so we see within the Malaysian 108 genomes. The majority of the mutation happen at non-structural protein 6, non-structural protein 12, nuclear capsid protein, and non-structural protein 3. Even though globally the highest mutation happen at the spike protein D614G, but in our local virus isolate, they are on number 5. So as equivalent to non-structural protein 12. So looks like our local isolate are mainly have mutation at non-structural protein NSP. So not really on the spike protein except for D614G. That's the mutation that happened in our country. So if let's say the vaccine or antivirals that targeting the spike protein, so I can say the effectiveness to in our country could be good if doesn't have any much mutation within the target proteins, spike protein. So I will little bit details about the spike D614G mutation based on Kober et al. Reported. It show the trend, global transition from D, the amino acid, aspartic acid at position 614, change to or mutate to glycine 614. So started February this year. So there is a trend that G614 mutation happen. So based on information that studied by Kober, so it shows that spike G614 variant become more dominant pandemic form within globally, and it shows that consistent increase of G614 variant at regional level may indicate a fitness advantage. So could be the survival of that virus is more prone for G614 instead of D614. And based on their study, G614 is associated with the lower reverse transcriptase PCR CT value. So lower CT value suggested that those patients with higher viral load. So, but their finding says that even though it's higher viral load but not associated with the disease severity, sometimes higher viral loads also have asymptomatic patient, so not really correlate between the higher viral load and disease severity. So that's their finding. So I think this spike D614G mutations is I think famous a few days ago, since a few days ago or since last week. Once our KKM DG stated about the emergence of these variants within the country. So that's why I will focus a bit on this mutation as example how we use the whole genome sequencing for the spike mutations. This is to show you where the the position of 614G in circle. So it's not really impact or change the overall main structure of the spike protein, but they somehow based on the Kober paper says that they losing a bit on the structure, so the binding affinity to the receptor at the our surface of the cells more easier, so they can penetrate the cells more faster, then can use our cells instrument to replicate within our cell faster. So that's why they produce a lot of virus within our genomes. Sorry, our cells. So that's why the viral load also high in patient with the G614 variant. Beside the mutation or nucleotide transition, so we also discover the deletion within the spike protein. So among two of our samples being sequenced, they carry the spike protein deletion. We have one of the samples which is case number 2 in our country that carry the 27 nucleotide deletion in N-terminal domain. And for one of other samples that we sequenced contain 15 nucleotide deletion at the subunit 1 and subunit 2 cleavage junction. So, but once we did the 3D structure for those spike proteins, it shows that those deletion are at the loop region for that particular protein. So it's not really impact on the overall backbone of the spike protein. So it's not really impact on the ribosomal, the receptor binding motif. So the deletions, even though they have the deletion within the spike protein, but it's still the functional of that spike protein to recognize the ACE receptor at the cells still there, still intact. So the deletion is not really impact on the how those viral infect our body. So that's a few finding that we discovered throughout the genome sequencing. So with this information, we can hopefully we can understand how those viruses react in our body and how we use those all the informations about mutations and so on. So how in order to combat or in order to try to eliminate those virus from patients. So I think that's about all I would like to share in this webinar. So as a take-home message for everybody here, so we can say that the whole genome sequencing of SARS-CoV-2 viruses contributed a very significant role in understanding the outbreak of the COVID-19 disease. So before I end the session, I would like to acknowledge a lot of our teams that involved in getting those the data and sequencing samples and so on from MDI itself, from Makmal Kesihatan Awam Sungai Buloh, IMR, and also MDI, Malaysian COVID-19 Bioinformatics Laboratory. Involve a lot of other institutions, UKM, USM, FGV, NARDI, and so on. So, and also MDI COVID-19 screening team. Anyway, thank you very much for listening. And if you have any questions, you can ask question at the Q&A panel, and also if you have any information that you need further information, you can email me. Thank you very much.
Thank you very much, Encik Mohamad Noor, for the very comprehensive talk. I think you have given a very good overview of all, about the virus, and also about the virus that you have sequenced from the Malaysian samples. We have a few questions, Encik Mohamad Noor. And I think more questions are coming now, but I will read to you the questions that I have noted. The first one was, has any group, either IMR or MGI or UM, sequenced the samples of the public cluster? So this is a question from Dr. Noor Shariza.
Okay, tablet cluster.
Yes, the tablet cluster.
I don't have the exact answer, but MGI doesn't have the samples because we are not screened for tablet cluster. But I know IMR and MKAK and UM also involved in screening the tablet cluster. So I presume that they have the samples, and later on they will sequence it. So at the moment, MDI doesn't have that samples. So I'm sure later probably IMR, MKK, and UM will share those tablet samples or genomes later on.
Okay, thank you, Encik Mohamad Noor. The second question, there are many questions, so if you can give a quick answer to all these questions, because I can see there are more questions coming in. So the second question is, this is from Professor Ravindran from USM Kubang Krian. Is there what method all the Malaysian lab employed for the COVID-19 genome sequencing?
Okay, in MDI, we have, in terms of technology, we have the Illumina technology platform. So we use a different approach. So we have two approaches, using the shotgun whole genome sequencing, and also we use amplicon sequencing based on Arctic Primer platform. So we have both platform for whole genome sequencing, random genome sequencing, and also amplicon. So and other groups that also using Illumina, but different machine, like IMR using the HiSeq, using the ISEQ, and also using the ISEQ, and a few group using the Oxford Nanopore platform. So they have a different approach and so on. So in MDI, we use MySeq platform.
Okay, thank you. There's another question, Cik Wano, from Professor Ravindran. Is there any difference in the virus strain between patient severity of the disease?
Based on our, because we don't have the clinical information for our samples, that based on our information, the viral load doesn't reflect on the disease severity. Sometimes we have a CT value very low, which is higher viral load, but those based on information that we have, those patient is asymptomatic, so doesn't have the symptoms, so and so on. So they still survive even though its viral load is high. So and they sharing similar close or very closely similar genome sequence within those higher viral load and lower viral load. So sometimes lower viral load also produce symptoms, influenza symptoms, so like cough and so on. So in genome perspective, really hard to say that the different type of virus that can have a different or severity of the disease. So
yeah, okay. Thank you, Cik Wano. Now there's another question. There's a question from Omar Ahmad. How different is the Malaysian SARS-CoV-2 compared with neighboring countries such as Singapore in terms of the genomic sequence?
Okay, it depends on the okay, let's say how those country get the first disease or first patient. So let's say because we already analyzed on our earlier first wave samples. So because of the relatedness of the Singapore samples compared to Malaysian samples, so these in terms of genome, it looks like very similar. But nowadays, because of our country got not only from Wuhan, sample from Wuhan, is get the samples from other countries, also from India, from US, from other Europe country. So of course the sequence is different within the country. So sometimes Singapore also have samples that originally from this country.
Okay, thank you. We have many questions coming, so I think it's good. Now the next question is from anonymous. No name was given. How long does it take to carry out whole genome sequencing of one sample?
Okay, I can say about on the perspective of Illumina platform that we use, which is MySeq. It takes, I think, for one samples we can process within a week, because it process, let's say for genomic DNA extractions and the library preparations and so on, take just a few days. And the sequencing just take about 19 hours. So roughly you can get one genome within a week. And not only one, actually, we can pull the samples in one run. So with the different target and so on, that's more technical. So like we run 24 samples within one run. So with the different barcodes and so on. So not only a single samples, we can run multiple samples in a day for sequencing. So it depends on the technology.
Okay, thank you. Now the next question, Cik Mohamad Noor, is from Noor Nadira. How is the nomenclature generated from the whole genome sequencing?
Okay, let's say for nomenclature that proposed by Roundabout, actually it involves a lot of genome analysis. So they did the multiple sequence alignment and so on, and then generate the phylogenetic tree and the clusters based on the clade phylogenetic tree, and they define as A lineage, B lineage, and so on. So B, and then they put some numbers to differentiate between those clades that from phylogenetic trees. That's one of the examples how they put the nomenclature from the whole genome sequencing. And the other groups using a different mutation. So they group based on mutations that happen and so on. So as I mentioned, Roundabout et al., I think the nomenclatures defined by Roundabout et al. Is well accepted now.
Okay. Mohamad, there's another question here. Have scientists identified the unique features of the virus isolated from cases worldwide?
Yes, definitely. Everybody try to look on the unique features. So because this RNA virus is very prone to recombination or mutations happen. So different continents, different locality, so they have some unique features for that particular sample. So definitely scientists try to find unique. That's why we have the classifications or nomenclature that we use to define what kind of features that our virus isolate have. So that's why we group based on the mutation, group based on the sequence similarity and so on. So but what kind of unique features in detail, so it depends on the numbers of samples being sequenced by each group.
Okay, thank you very much. Now there's a question here from Saila Ismail. Recently in the news, and I think you mentioned about this also, it says the Siva Ganga cluster may be caused by the super spreader D614G strain.
Yeah.
So any updates on this?
Okay, we don't have the sample. I assume the KKM, because the samples is handled by KKM, I assume IMR will come up with the sequencing data very soon on the Siva Ganga cluster. So I cannot comment so much because we don't have the samples. If we have the samples, then we can do the sequencing and let everybody know what kind of mutations that those clusters carry. Okay, thank you. Now,
Muhammad Effendi Abdul Wahid has another question. Were the virus attached and colonized mucosal membrane of the upper respiratory first or enter the bloodstream at any sites of the body?
On the basically what we know that those virus using the spike protein attached to the mucosal membranes of the upper respiratory parts of our body, and in terms of entering bloodstream, I'm not really sure on that because mainly they are infect the our respiratory systems. So in terms of bloodstream, I'm not really sure how they go in. Actually.
Yes. I think we need to ask the virologist.
The virologist, no, yeah.
Yes. But this is also very interesting because I have the same thought before. If the virus attached to any parts of the hemoglobin, to travel from one organ to another.
Yeah.
For example, from the lungs, to the other, to the brain or other organs to cause, systemic infection.
Yeah.
So how do the virus travel? The virus, do they attach to the hemoglobin? So if they do attach, then we see that people or patients who have problems with their hemoglobins or born with abnormality in their hemoglobin,
yeah.
Will have, the virus will have problem, to spread within the body of this, this individual. So anyway, that's my personal thought, and I share the same thought also with Mohamad Effendi. There's a few more questions, Encik Mohamad Noor, if you don't mind, boleh ke?
Can. Sure.
Okay, this is from Anonymous. What do we know about the potential causes of the mutation? Can individual genetic profile influence the mutation? This is again, I think, another question which I think I share. Can individual genetic profile or the patient's genetic profile influence the mutation or the spread of the disease or severity of the disease?
So far, I didn't come across yet people that study the genetic profile for patients and the type of mutation within the virus. So I cannot comment much on the influence of the genetic profile. But again, as mutation happen in virus because they are adaptation. The virus need to survive, so they need to mutate or to adapt on the stress environments or to make sure they are survive. So a lot of other factors involved in mutation. So it could be environmental factors, could be the immune systems that from the patients and so on. So the virus need to mutate in order to camouflage our immune systems and so on. So in terms of genetic profile influence, I've not really come across any study yet on that.
Okay. This is the last question from Azwan Awang. We know that some animals like dog, tiger, lion, mink have been infected by the virus elsewhere. Could you please comment on the possibility of infection to livestock? Mutation on S protein, spike protein, seems a threat.
Okay. How this virus recognize those receptors, different receptors for every cells, different animals and so on. So it could be mutation at the spike protein can be a threat but somehow those SARS-CoV-2 that infect human so far no reported that can infect livestock so far, but some other reports that they can. I read a few reports previously that the cats that those patients sit together with the patient, COVID-19 patient, also in fact also have some kind of influenza-like illness to cat. So on the livestock, I don't have at the moment. So it can be the mutation of the spike protein can be a threat to livestock also. So because coronavirus not only infect human, so it can infect animals and other organisms. So we found coronavirus in dog, cat, and others.
Yes. Okay. Okay. So we actually, we have exceeded the time allocated for us. It was supposed to be 1 hour, but it's I think more than 1 hour now. There were very good responses, participation from the from the attendees. So that is the last question from the participant. But before we end, can I just ask one burning question?
Yeah.
This has been, in my mind since the beginning of this pandemic. Now, this SARS-CoV-2 virus, there has been also, there has been also people who raise up this issue of this pandemic or this virus being, being manipulated, and also, it was meant to be a biological weapon. What do you comment? In fact, personally, I've been asked this question, so I'm very interested to know your opinion on this. Whether you think it is it started as a biological weapon, or somebody, somewhere, started this as a biological weapon.
A very tough question to answer actually. I'm also there is a rumors that people study those coronavirus for biological weapon and so on. So, but based on evidence that discovered by scientists, shows that those virus is naturally occurred from bat to pangolin and to human. So basically the coronavirus is actually everywhere. In mostly the reservoir is in bat. So sometimes, among the scientists also have a nasty scientist that try to play dirty games and so on. So I'm not sure this virus came from or propagated from certain lab for bioweapon and so on. But based on the threat, it can be used for bioweapon because it's like pandemic right now. So I cannot comment so much on the bioweapon because I don't have any idea beside that is naturally occurrence from there's a evidence naturally occurrence from bat to pangolin and human. So beside that, I don't have any information.
Yes. I think this is a question which I think we will be in our mind. Many of us, we are thinking of this, and nobody knows for sure, the right answer. So, so Encik Mohamad Noor, one of the participants has one last burning question. Muhammad Afandi Abdul Wahid. He has asked, I remember, 2 questions, and this is he said this is his last question. So, do you mind we take this because I think it shows that this is a good participation from the attendees. Now, his question is, in your opinion, can we focus in stimulating antibodies at the local mucosal sites of the mucosal lymphoid tissues rather than focusing on the modulate immune response on systemic immune system?
Okay, first of all, I'm not an immunologist, so but I can say in my opinions that because the mode of entry of these viruses actually attach to the mucosal sites of the tissues. So I think the best, or not really the best, the one of the options that we can try to stimulate the antibody within the mucosal tissue is one of the strategy that it can be done instead of modulate the immune response, because on the systemic immune system. So we try to close the gate at the mucosal membranes instead of gut everywhere. So it depends on the how we modulate those immune response. So I think, yeah, it can be done, one of the strategy, but sometimes, yeah, the immune, sometimes the selectivity of those antigens that need to trigger to produce the antibodies locally or just certain areas a bit challenging in my opinion.
Thank you, Encik Mano. Encik Mano, do you mind to stop sharing your slide? Because there's a request from one of the participants whether, you know, they can see the face of the speaker while answering questions. But anyway, that was the last question. But still, I think before, you know, we say goodbye to everyone, perhaps, you know, maybe they would like to, you know, to have a look at your face when you say your, you know, your final words, and make, you know, maybe, you know, give another or reinforce your take-home message. Can you stop sharing? Have you stopped sharing?
Yeah,
oh, okay.
Sharing my screen already.
Great. Okay. So would you like to have, you know, like a final, give a final take-home or reinforce your final take-home message to everyone? You know, it has been a very good session. Everybody, I think, I'm sure every one of us, we have enjoyed this. We have more participants than we expected. We expected about 60 or 70, but I can see here that 94 people registered for this webinar. So it's way, you know, beyond our expectation. And as you can see from the questions that has been posted, everybody, I think, yeah, enjoyed the session, you know, and they wanted to know more about, you know, this virus. So Encik Mohamad, do you have any final words or any last take-home message?
Okay. First of all, thank you very much for those listening to my talk, and hopefully you can gain some things from what we have done in MGI and the rest of our teams here. Again, during the pandemic, we have to study because of this virus is novel. Still don't have information, a lot of information. We need to do the whole genome sequencing and so on to understand in detail what kind of the control mechanisms on the infections and so on. So the blueprint of the genome of the virus. So it's a good way to understand the pandemic of this virus through genome sequencing of those unknown viruses. So again, thank you very much, and stay safe, wash your hands regularly, and wear mask at the closed space.
Okay.
Thank you very much.
Thank you very much, Encik Mohamad Noor, for the excellent talk. We have all enjoyed it, and you know, thank you also for the, you know, the you know, the kind advice. And I think we thank you again for this talk, and you know, and I thank everyone, all the participants, for participating and for bearing with us. You know, we have exceeded the time, but I can see that, you know, there's still many participants still, you know, online. And I thank you, Encik Mohamad Noor, from all of us, and thank you everyone for participating. And I hand over the floor to Dr. Azzreena. Silakan, Dr. Azzreena.
Okay, thank you very much, Prof. Zilfalil and Encik Mohamad. No, very interesting sharing and finding from MGI and other institutions involved. Okay, so to all participants, thank you for participating in today's webinar. On behalf of the society, we hope to see you again in other events organized by PGM. Okay, so to PGM members, I would like to inform that we have 25th AGM later, which will be started at 10:45 a.m. Okay, see you all in the AGM later. Okay, so that ends the webinar. Thank you very much.
Okay, thank you everyone.